InContext, and it allows researchers to perform gene set enrichment analysis while also incorporating some additional experimental context to ask some more targeted questions and address more targeted hypotheses. So in the past 20 years, gene expression technology has allowed us to measure how the thousands of genes in our genome can change, can increase or decrease in their expression following certain biological changes. And this could be over the course of normal development, or in disease states like cancer, or under any other sorts of alterations. Our lab happens to be interested in the pharmacogenomics of bladder cancer. So a lot of the examples that I'll be talking about today are most relevant to the experiments that we perform in the lab. But it's important to note that anything that I'm talking about can be applied to those other contexts as well, really any sort of gene expression experiment where we're comparing these two sorts of groups. So a typical transcriptomics experiment that we perform involves two groups of human cells, one case where they've been treated with a small molecule drug, often in our case, and the other group has been treated with an appropriate vehicle control. So we measure gene expression in these two experiments, and then we perform differential expression analysis. And usually this analysis is summarized then in terms of a ranked list of genes. And so here you can see genes that increase in expression with drug treatment at the top of the list, genes in the middle showed no change under the drug treatment, and then genes at the bottom of the list decreased in expression. So we have a lot of different tools in our bioinformatics toolkits to be able to interpret the potentially hundreds of changes that we could be seeing when we do an analysis like this. And perhaps one of the most widely used tools is gene set enrichment analysis, or GSEA, which has been cited almost 15,000 times now since it was first described in 2005. So I'm sure many of us in this room are very familiar with GSEA. I'm going to give a quick overview by means of introducing our method, which is intended to complement it and extend it a little bit further. So GSEA is based on a key observation in genome-wide expression experiments, which is that changing biological conditions can cause modest but coordinate changes in gene expression levels for functionally related sets of genes. And so GSEA leverages these predefined gene sets that can be hosted, for example, in the MSigDB database as basically to ask the question, are these biological pathways or processes altered in my experiment? So like I said, the MSigDB database has these gene sets available. For the purposes of keeping everything in this talk simple, I'll just be talking specifically about the hallmarks collection, which contains gene sets that are mainly annotated to what I like to think of as the textbook pathways, lots of kind of canonical pathways. But it's important to note that the GitHub pages and Synapse pages that I'll show at the end do have the results for all of the gene sets in MSigDB run as well. So to identify pathways in our experiment, we measure gene expression, we perform differential expression analysis, we get our ranked list of genes, and then we use that as input to GSEA alongside our desired gene set. And really the purpose of GSEA at this point is to give us a score for every gene set that we've asked it to analyze. So in order to do this, we've now taken our ranked list of genes as input, laid it down here on the bottom, and then said, I'm going to look for hits now that are in this gene set along my own ranked list. So the question that we're trying to address here are are these TNF alpha related genes tending to fall towards the top or the bottom of this ranked list? And this is what we refer to as positive or negative enrichment. So another important part of what GSEA provides us is this overall enrichment score, which we can see is the peak value as we're walking down this ranked list and seeing our hits. But importantly, GSEA also provides a p-value for this enrichment score. And this is important because without it, we wouldn't have very much context for the enrichment score itself in terms of how significant that really was. So to estimate the significance of the enrichment score, GSEA uses permutation testing, which means it compares my enrichment score that's been computed on my real data with the TNF alpha gene set to an empirical null distribution of enrichment scores. And GSEA offers two different methods for computing this empirical null distribution. The first option, and the more statistically powerful option, is to shuffle the sample labels as part of the permutation, recalculate the enrichment score for all of these, and that gives you your empirical null. Unfortunately, and I'm not going to go into this just in the interest of time, but this method does require and is recommended to be used only when you have 7 or more samples per group. So I'm not going to be talking about this one. Really what I'd like to focus on is permutation strategy number 2, which is permuting the gene sets. This algorithm in GSEA is called the GSEAPreranked algorithm with the gene set permutation. And specifically, this applies to a lot of the experiments that we perform in our labs. And now, based on what I've seen in publicly available gene expression data sets as well, I would have to say that many, if not most, of the gene expression experiments we see have fewer than 7 samples per group, at least the ones that I've been looking at. So in this situation where we're permuting the gene sets, we start with our original TNF-alpha signaling gene set, and then what we do to create the permuted gene sets is we randomly select genes from our overall ranked list that we started with to create gene sets that are the same size as the TNF-alpha gene set that we started with and we're analyzing. Because it shuffles the gene sets in this case, it does break possible correlations between genes in our data set and genes in the gene set. So it is a weaker statistical test overall, but like I mentioned, it is relevant to most of the experiments that we're working with. So what happened was when I joined the lab, one of my first projects involved performing a lot of differential expression analysis on different bladder cancer data sets that we had, different experiments that we performed. So for example, you can see some bladder cancer cells treated with cisplatin, a DNA intercalator. But what happened was I was doing these different experiments, and then I would take my results, my GSEA results, and compare them to results that I was seeing in publicly available databases as well. So I would find another publicly available experiment, in some cases different cells treated with the same drug, and look at those GSEA results, or different cells treated with different GSEA results. And the more I did this, the more I started noticing trends in the pathways that I was seeing positively or negatively enriched. Well, being in a pharmacology lab, we often think first about drugs having a very specific target and therefore a very specific effect on the cells. So we became curious about this and wanted to investigate further. I specifically was interested in looking at this actually in a broader context as well. So when we go back to the original permutation strategy for that gene set-based permutation that I was talking about, what we realized is that it's making the assumption that all of the genes have an equal chance of landing in one of these permuted gene sets, or that really all of the genes can fall anywhere in the ranking. So when we looked across lots of publicly available data sets, we decided this would be a good thing to investigate. I started by just going to Geo and finding all of the data sets that I had available to me that used the same microarray just to keep the gene annotation straightforward at first. All of these data sets had human cells, at least 2 replicates per group, and treated with a small molecule inhibitor or an appropriate vehicle control. And really the goal here was to arrive at the most diverse set of experiments possible so that we could test and see whether these pathways really were being positively enriched across a wide variety of conditions. So in total, 21 tissues and 129 different small molecule compounds were represented. I took all of these data sets and performed the standard differential expression analysis that I had been doing to arrive at 442 ranked gene lists. So the first thing I did when I had these ranked lists was just plotting the average rank for each gene across those 442 data sets. We can see that this distribution centers around 8,000, which is about half of our ranked list, so that makes sense. And I pulled out the genes that had the highest and lowest mean gene rank as well, just to investigate. What we can see is that the gene that was most frequently increased with drug treatment across this wide variety of experiments was MAF, a transcription factor involved in detoxification and response to oxidative stress. On the other hand, the gene that was most frequently decreased in response to drug treatment was cyclin E2, which is highly expressed during the G1 to S phase of the cell cycle. So both of these results are consistent with what we might expect from cells that have been treated with a small molecule drug. But to test, and you can see that these distributions are pretty striking as well in terms of the number of experiments that they're increasing or decreasing expression in. So to test whether this was really a few troublemaker genes here versus more of a genome-wide effect, I now simulated 442 randomly ranked gene lists. And we can see, based on this random simulation, that actually over 25% of the genes are fitting one of these profiles. So they're being increased or decreased in expression more than we would expect by average. So it seems likely that this could be affecting GSEA results, particularly if genes within the same pathway or process are falling to the same side of this heavier-tailed distribution. So to test this further, I took the 442 experiments that I curated, ran them all through GSEA, and tallied up the results. So what I'm showing you here now is for the 50 different hallmarks gene sets, the proportion of those 442 experiments that have that gene set positively enriched, significant at an FDR-adjusted p-value of 0.05. And to take an extreme example, you can see that TNF-alpha signaling is positively enriched significantly with GSEA in 53% of these experiments. And we can see a similar trend actually with some of the negatively enriched experiments E2F targets and other proliferation and cell cycle related pathways are consistently downregulated and negatively enriched in these wide variety of experiments. So what we really wanted to know was, how could we leverage this information to actually better inform our own experiments? So in other words, given that we're seeing these big trends across the data sets, if I see TNF-alpha positively enriched significantly in my own experiment, great, that's a biological result, and we do really want to know that. But given now that we know that that same result occurs across many data sets with many drugs and many cell lines, can we use that information to put our own results in context? So how can we use this information to better understand our own experiments and potentially ask more targeted questions? So this was really the motivation for developing our method, GSEA InContext. This figure from our paper summarizes the original GSEA pre-ranked algorithm, and the key part to focus on here is really how the permuted gene sets are created. So by assuming that any gene A, B, or C can fall anywhere in our ranked lists, when we create our permuted gene sets, those can include essentially any gene at random to create our empirical null distribution. However, the GSEA InContext approach takes the same inputs before with two very important differences. Now, in addition to our ranked gene list that we give it, our collection of gene sets that we'd like GSEA to analyze, we also provide a set of user-defined background experiments that we use to actually inform this distribution. We estimate a density distribution using the beta binomial for each gene. So each gene has its own specific density estimate that we then use to inform our permutation strategy. So when the gene sets are created, they're informed by this distribution. So the take home here, really, aside from the permutation strategy, is that if these genes are in my gene set, and I have in my experiment gene A is decreased, gene B is increased, and C falls somewhere in the middle, that gene set is not going to look very significant in my experiment because it's very similar to all of the background experiments. The goal here is really to identify what's unique to my experiment as opposed to others. So here I'm showing you the same histogram that I showed originally with the large proportion of experiments that had the TNF-alpha signaling enriched over here. I'm just going to leave these points here and compare them now to the points, the proportion of experiments with that gene set positively enriched when we use GSEA InContext. And what you can see is that now here in the lighter pink color, the proportion of these gene sets when I use, I skipped a step. To generate this plot, I took each of my 442 ranked lists now. I took them one at a time and then ran GSEA algorithm using the whole other set of the 441 as the background. So now what we can see is that the method is doing what we would expect it to do. So by using this whole background set of experiments and comparing each one individually, we see that the overall proportion of these experiments has decreased, and you can see the same thing in the negative case. So here in the dark blue, these were the original proportions of the experiments that had each of these gene sets negatively enriched, and that number has come straight up to 0. So really the ones that are the number of experiments that are still having these different gene sets positively or negatively enriched, it's more unique in that experiment than it would have been in the entire background. So I wanted to kind of highlight now two specific examples because we really think the power of this approach in terms of the example that I just showed you using the entire background set of experiments, the power of this approach is that we can target the background set to really address a question that we're interested in. So one common task in the field of pharmacology is to understand the effect of a small molecule drug on cells. In the case of this publicly available experiment, I grabbed some breast cancer cells, MCF7 cells that had been treated with both dexamethasone and estradiol, and compared those to MCF7 cells that were treated with an appropriate vehicle control. And using, again, the hallmarks gene set, we can run the original GSEA pre-ranked algorithm, and you see a number of pathways are either positively or negatively enriched significantly by GSEA. But to illustrate how GSEA InContext can leverage a background set of experiments, I compiled a set of these 22 other experiments using MCF7 cells, but treated with estradiol alone. So now here the comparison that we're using in GSEA InContext, which is this orange box here, is saying how is this experiment where we treated with dexamethasone and estradiol different than when we use the same cell line but treat with estradiol alone. And so GSEA InContext has now picked up a subset of these pathways that are predicted to be more significant here in the case of dexamethasone treatment as opposed to just with the estradiol alone. And so these are really interesting results. Dexamethasone is a corticosteroid, and so it is known to affect androgen response. And another interesting result that you may see here is that the complement pathway was found to be significantly negatively enriched by GSEA InContext, but it would have been missed by GSEAPreranked. And what's interesting about this is that dexamethasone is actually known to be a transcriptional regulator of some of the genes in the complement gene set, and was also shown to downregulate complement in immune cells, so this may be a good area for future research in breast cancer cells. And so the final experiment that I wanted to show in this example are these, is another cell line that's treated specifically with a peptide that was designed to inhibit Notch. And so the challenge here really is saying, okay, if we run GSEA with our hallmarks gene set again, I'm showing you now on the x-axis the p-value for those GSEA pre-ranked results. And what we notice that here in the yellow and green, all of these things significant in GSEA pre-ranked, we don't see Notch signaling. When we compare those results to our results with GSEA context, we do actually see Notch signaling. So how did I do that? Well, I used a targeted background set of experiments that were, again, lymphoblastoid cell lines, same as the original experiment, and they were just treated with any other drug. So now the comparison in my question is, what is different about treating these cells with a Notch inhibitor as opposed to treating them with any other drug? And here we see that Notch, the Notch signaling pathway is identified by GSEA InContext. And so just some conclusions and closing remarks about this. We compiled a large set of gene expression experiments, identified trends in terms of the gene rankings across them, and how the non-random distribution of gene ranks may be affecting GSEA results. So to address this, we developed a method, GSEA InContext, which accounts for gene expression patterns within a user-defined set of background experiments and leverages that information to produce more targeted GSEA results. So really, what I hope to have shown you in these examples, too, is that we can evaluate small molecules with known targets. We can evaluate these more targeted contexts and get interesting answers. So overall, we hope that GSEA InContext provides researchers valuable insights that complement standard GSEA pre-ranked analysis. And with that, I'd like to thank my lab at the University of Colorado Anschutz Medical Campus, our collaborators. All of the data and results are available on Synapse and GitHub. And I would also like to give a shout out to the University of Colorado Computational Bioscience Program. We have a booth at the career fair tomorrow, so please stop by and see us at booth 6. Thanks.
Hello, it's really the hot topic, thank you. I have a question. So when you work with the geo and so many data sets that are there, the different labs use different procedures for the normalization and for batch effect correction. Mm-hmm. How do you address these issues in your method? Mm-hmm. Thank you.
That's a good question. So I used the raw microarray data in the form of cell files, and I analyzed them all with the same bioinformatics pipeline. So that was using just some standard R packages like AFI, Lemma. I did RMA normalization. So in the paper, we talk about the consistent normalization strategy that we used, but really starting with the raw cell files allowed us to be able to use that same pipeline and using only experiments with the same microarray.
So first, that was an incredibly clear talk, so thank you. So one comment and one question. So the comment is, I don't know if you noticed when you read the literature, but at least to my knowledge, the original GSEA paper never introduced, right, it's a 2003 paper, and it never introduced this concept of pre-ranked, right? So this was like an addendum to the method, right? And so somehow this managed to slip through, I feel like, and as a community, we all embraced single sample GSEA, right? As a method. And so you illustrate well the problematic nature of that as a statistical method and the fact that it is underpowered and sort of produces false positives. So that was great. So my question is sort of like on the in-context side, right? So you have two different comparison conditions, right? What if they each follow a different expression distribution, right? It seemed to me at least that the null model doesn't include that assumption, or am I wrong?
Oh, I see what you're saying. Yeah, so that's a good question. So when I'm making the comparison, when I'm using a background set of experiments, what I'm using is already the differentially expressed genes from that comparison. So all of those ranked lists are already in terms of drug treatment over control. So really the distributions that we would be comparing in that case are the distributions of all of the differentially expressed genes.
Certainly, but I feel like there's this implicit assumption, but maybe I'm missing, and maybe we could take this offline too, that there is no change in the, right, what you're actually wanting to see, right, is that there's a concordant change in expression within a pathway, right? That's what the test is about, right? And now, if different genes operate on different scales, which is one of the take-homes that I was taking from, and we're ignoring this completely when we're testing for enrichment, right? Then, like, what if this scale, right, changes between conditions, right? So a certain gene under normal condition, right, operates at a certain scale, and under a different perturbation condition or drug treatment, right, it now moves to a complete different scale, right?
Yeah, I think that's fine with the method that we have. So we use classic GSEA rather than weighted, so the log fold change values aren't taken into account. And the only thing that we use those ranked lists for are actually the genes themselves, so just integer values for the ranking, so not the fold change values. So it's really just the relative ranking of those genes in the list are what that density distribution is based on, not the expression of the genes or the differential expression.
Okay, thank you.
Thanks.
Very great question. How big does the background set have to be in order for your method to work?
That's a great question too. So what I wanted to show in my examples was that I can get down to 20 or 10 different experiments and still use that as a viable background set. So in the paper we do some subsampling analysis to show how the results stabilize with fewer and fewer in the background set. But being able to get down to 10 is pretty great, I think, because you can find publicly available data sets, hopefully that many.
And what's the ideal?
The ideal would just be the bigger the better, always.
Thank you.
But,
when you put together the data of the different labs, did you consider the concentration used for the treatment? Because particularly for the kinase inhibitors, depending on the dose that you use, the gene expression changes.
That's a good question too. So in some of these cases, the gene expression experiments had measurements at different doses, and so all of those were actually included in this giant compendium that I had. So I didn't take into account what the relative concentrations were. I just used everything in the background set, again just trying to get a diverse set of experiments. So I think that's really worth looking into, and perhaps if you wanted to use a background set that was very targeted to what you were looking at, you could take that into account. But really what we were using was a generic background set for what we were using.
Thank you.
Thank you.